Hello. Yes. So we will be continuing with the previous NGS workshop, in which the last part was about the use of advanced containers for metagenomics, and after that, the molecular dynamics and structural biology hands-on will begin. So I'm very much thankful to my colleagues for taking a very extensive session on VCF analysis pipeline, which has made you much more familiar with understanding Bash scripts and Linux commands and how they are executed. So for this tutorial for metagenomics, I have created some Bash scripts. So I will shortly share the tutorial with you. So I hope everybody is able to log in. Already logged in? Just a minute, I'll just share the links with you. These are some Google documents I have. So I'm sharing the link for the tutorial for this hands-on. So we'll be looking at the use of advanced containers. Till now, whatever you saw was all about using basic containers. So if you'll go back to home, so here we begin with the use of containers, and you go to images, and from here you select whichever container you want to use by clicking on the launch button. Now going back to this page for instances, here you'll see another button that says create advanced container. So we'll be using this advanced container. So before that, I will just take you through the tutorial that I have shared on the chat box. You can download this tutorial and follow it in future. It is not available on the ICE. I have just pasted it in the chat box. So this tutorial will show you the steps for running your own Docker image, and which has been created using a Jupyter base. So this is one example case I'm taking, which was created using Jupyter for compiling the Docker image. And this image has got CQC, is named CQC, and it includes sequence QC tools, which you have, which you are almost familiar with in the previous hands-on session. And the aim of this tutorial is to make you understand how Jupyter Lab interface can be used, and you can use the tools in the terminal from the Jupyter Lab. So first we will start by creating this container. So you go to containers and then to instances drop down. So here in the container button, you go to instances and this page will appear. And then you have to click on create advanced container. In this advanced container, in the tutorial, I have mentioned what all you have to fill in the specifications form. So this is basically giving you all the fields to be filled for specifying what all are your requirements. So now I'm typing in the job name. So whatever has to be typed is in blue font. Copying it. Sequence QC. Now this is the container I have hosted in the Docker Hub in my own login, which is suchp. Here, therefore, this has to be copied as it is. So we are pulling a container which is outside of Ice Cloud. which is on the public Docker hub. Then memory allocation. So here we are selecting 16 GB of RAM. In the previous session, your RAM availability was increased. So this will work only if you have got 16 GB or more RAM available or allocated to you as a user. We are selecting two CPUs. Here it's two. Then this is the container port. And this is the command which will actually help run the Docker. So you have to paste it in this field. After that, we still have to mount our drives. So till now, whichever images you have been using, these drives were already mounted on them. Here, this customization is available where you click on this blue button to add these drives. And in this drop down over here, you will see which all folders can be mounted. So public data is the public data folder present on ICE. So I'll just open another ICE here and show you which folders we are mounting. So in your storage and file drive, this is your public folder, and whatever is present over here is going to be mounted by selecting this public data over here. And for whatever is present in your home directory will be mounted from the second option here, where I'm selecting private data. So whatever is your username, you have to select private data for mounting your own drive, home drive. Now the mount path over here has to be /drive / and small case public. Okay, no /. And for private data, again, I'm typing drive / and instead of typing my own name over here, I am keeping it as user. So this is very important since I want everyone to use the same path for this tutorial. You should see that your private data drive which is mounted on this Docker will be user. So drive user. So you don't have to type in your name every time. It should be just drive user. So you can see that in the tutorial also I have mentioned what is the volume mount path and what is the path to be typed in. So for private data, drive user. Only then this tutorial will work for you. After typing this, you have to click the create container button. Okay, so here, just revising, we have mentioned the job name, image name, which is going to be pulled from outside, your memory, your CPU, your container port, the command we have copied from the tutorial, and the paths to be mounting your public data and private data. Here, if you want, you can also add another field for shared folder. But right now we are not using it, so I'm not showing it. Now I'm clicking on click container, create container. So now it is saying successfully created pod. Okay. And here you can see the image name CQC. When it was created 1 minute ago, 2 CPUs have been allocated and 16 GB of RAM has been allocated. Now I click the refresh button. We can see the pod is running. Now you scroll to your right under this actions column, click on this 3 dotted options and click on connect. Now it will launch the Jupyter notebook into a new tab. So this was our previous tab. After clicking this connect button from here, we have come to this Jupyter Lab. Now in this folder, you will see here instead of your username, since we typed there specifically to rename it as user, instead of your own name, you will be saying user over here. So if you'll open this folder, whatever was your previous tutorial, all those files are over here. So suppose your previous user's name was Tina. So here it was Tina folder, and inside that all these files were being generated and accessed from. Instead of that, now we have renamed it as user. Now for this tutorial, we are going to use the public data. So I will just show you where it is located on ICE. So you go to ICE workshop. In the ICE workshop, NGS analysis. In the NGS analysis, in the ICE workshop, CQC data. And here is your test data that we will be using in this tutorial and the scripts. So these are the bash scripts that I have already created so that you don't have to type in all those commands which we saw in the previous hands-on session for every command. So for example, fastqc.sh has got, okay. Um, the path to this folder is, I have got a query right now. So I'm just showing you again, go to storage file drive. Okay. So then you go to public drive. Okay. In the public drive, you scroll down after clicking on public drive. And here you'll see ICE workshops. In the ICE workshops, again, you scroll down. You will see different tutorials that we saw. In the morning, we saw NGS analysis. Now we are going to look at CQC. So I'm opening the CQC folder, again scrolling down. Here I can see two folders. The test data has the raw data and scripts has got the scripts for running. So I was showing you the example for FastQC script. So we are not going to type in all these commands, but we'll be running the bash script directly. So I'm explaining it to you before. So firstly, this has got multiple Linux commands which will run one after the other. So whatever has a hash sign, that line is not a command, it's a commented line. Whereas wherever there is no hash, that is the actual command that will be executed. So this is mkdir -p drive. So this is going to create a new drive in your folder. So here, since it is inside user, it will create a new folder named CQC out fastqc. And it will be running the analysis directly by without copying the files straight away from the public folder itself. So these files, fastq files, are already in the public folder, and the output will be generated in the user folder in your fastqc folder that was created using this command. So now going back to the tutorial. So after you have created this container, the next step is click the refresh button and connect. And we saw how to open the Jupyter notebook. Now going to the 10th step, open a new terminal by clicking the terminal icon. Okay. So go to your Jupyter Lab tab. We saw the folders, how they are. Now I'm scrolling down. So this is the launcher tab that is present inside the Jupyter Lab interface. When you scroll down this launcher, you will see this terminal icon. Is everybody with me? Has everyone found the terminal? Sorry. No, right now, have you opened your Jupyter Lab? Yes, so it is already in the user folder. So here, whatever, okay, I'm just repeating it for clarity. I'm closing this once again. So till now, till here, everyone has followed how to generate this container, right? This is just like previous containers that you have, and it is running. And from here, you have to connect it. When you click on connect, it will open a Jupyter Lab in a new tab on your web browser. Is everyone following? Yes. So once you open this, our mounting paths were the root directory in which we have mounted the public folder and the user folder. So this user folder is actually your home directory, and this is the public folder. So whatever data we'll be using is in the public directory, which we saw even from the ICE web page. And in this, since this tutorial has multiple tools for sequence QC, firstly we'll see what all tools are installed. So have you all found the terminal icon? Yes. So I'm clicking on this terminal icon and it opens a new terminal. So this is just like a Linux command line interface, which you were using in the previous Docker images. So here also you can type pwd, that is present working directory, and check which directory you are present in. So right now I am in the drive. Now I can change my directory by saying cd. Now instead of typing my name, I have mounted my directory using user as my name, cd user. Uh, I think there's some query from the online participants. I'll just go back to the meet. Um, can you please mention flow here? Okay. So Abhijit has shared the tutorial link once again. So for all those who have joined right now, please open this link. Right, we are going through this tutorial for using advanced container. I'm just repeating whatever we did till now. So we have created an advanced container using container instances option here. In the container instances page, you will see a create advanced container button. By clicking on this button, it will open a form where you have to fill in all these fields. So we have filled in all these fields in the container form. And after that, we click the blue plus button for mounting the private and public drives. So we have mounted public drive and private drive at these paths: drive public, drive user. And after that, we click on the create container button. Here, if you can see, this was the form that we filled, and we clicked on the create container button. After which the pod starts running. After it starts running, we connect to the pod, and after clicking on connect the pod, it will open a new Jupyter notebook. I hope everyone is following. Yes. So after that, we launched the terminal, and in the terminal, we are seeing where our drives are. So we typed in pwd to see where we are presently located. It's in the drive folder. Then we go to cd user. After doing cd user, you will see whatever files you have generated in your previous tutorial, right? So this was your actual directory previously, which was named with your own username. Instead of that, we have renamed it here while launching this image as user. So going back to the tutorial, these are the tools that we have installed in this single containers. So previously you saw every command had required opening a separate container. But whereas in this container, we have installed all the tools in single container. So you need not close the pod and reopen a new pod this time. So now I'm just copying this fast QC command from here to check if it is installed. So here I'm saying paste. Okay. And click enter. You can see fast QC is installed. Going back to the tutorial, fast P command I'm copying. Copy, paste, enter. Fast P version 0.24 is installed. Similarly, seek kit. So seek kit is another sequence file exploration tool. Okay. So seek kit. Okay. So this was the help that was generated here. The version. So whenever you are using Linux command, it will actually print if you're typing in wrong command, sometimes it generates the help for it. So there was no dash dash required before the command for version. So it printed the entire help. So you can see ckit version is installed. And for metagenomics, I have also installed centrifuge over here. So centrifuge dash dash version, enter. Okay. So next, we will see. So you will see something like this after you have completed running all these commands I have mentioned above. And now you are ready to perform sequence QC on your data. So coming to the metagenomics tutorial, the first step of any NGS pipeline is to assess the quality of raw FASTQ files, which is important for removing low quality reads and increasing the accuracy of your downstream analysis. So this particular container has the most popular tools installed in it for assessing your data. And after that, we will be using the centrifuge for taxonomic classification. So now for running your scripts, firstly, we are starting with the quality check of raw sequences. Now, from wherever you are present, since we are copying the complete path of the scripts folder, you will move to your directory will change to scripts folder from where we will be executing all the scripts. So I'm copying this right now. Going to the terminal in Jupyter Lab and I'm pasting it here. Enter. Now if I'll type ls, I will see there are 4 scripts present in this public folder that we will be using for this tutorial. So first 0th script is fastqc. So to run this script, you just need to type bash before the script. So this 0_fastqc is basically just a text file which contains the Linux commands that you have to execute. So to run them in together, you create a .sh extension for it to be executed using bash. I'm just copying this and I'm pasting it here and click enter. So now you can see it is running fastqc for the samples. Fast QC has completed. Next, you go to trimming of poor quality reads using fast P command. So I'm copying it and pasting it here. Bash 1_fast P. Enter. Fast P running has completed. The third is the centrifuge command. So we will start running centrifuge. Enter. Now this centrifuge is basically using a database from the public directory. So I will just show you over here. It is using DB centrifuge PHV. So PHV is prokaryote, human, and viruses. So it contains indexes for prokaryote, human, and virus genomes, which it will which will be which will be used for annotating the FASTQ reads that we are having in this tutorial. So now sequence centrifuge has completed its run. The next is, since it has already completed, we will skip the fourth step and we will go to fifth step, that is bash k report, control C. And in the terminal, I'm pasting this bash k report. And in the script itself, I have kept a multi-QC command, which is why it is going and fetching all the results that are present in your output folder, and you get the complete report generated, which is saved over here. So now I will just, since we have almost completed all the commands over here, before we go to visualizing output, we will just see where all these scripts are again and how they were executed. So the scripts were in the public folder. We are here, drive CQC scripts. So drive ICE workshop CQC scripts. So previously I showed you how to browse from ICE to this scripts folder. Right now I'm showing you how to browse to this folder within your Jupyter lab. So you need not change to the ICE interface. So here itself, you can see fast QC commands that were executed. Fast P, I'm just double clicking these files and they are just opening in the panel besides. There's a centrifuge commands here. So since these are much complicated and would take much longer to run individually, I have created these bash scripts for the time constraint. And so this was the last codes that we have executed in this. We have executed multi-QC command, which generates a complete summary of the output that for the files that you have generated using first three commands. So since our outputs from the previous commands was stored in the out folder in the user drive, here I have given the input as drive user CQC, for which the output is again in your drive user CQC out folder. So now here, if you will go to your user folder, there's a CQC folder which is created. Is everyone following? Yes, in this you have the out folder. In the out folder, you have this HTML files. So all these folders are created using these scripts over here. And you can just right click on the HTML file and select Open in New Browser tab. When you click this, your entire multi-QC report is visible, where you can see there were two samples that were processed. Their FASTQ outputs are summarized over here. The quality outputs are summarized. So entire QC has been summarized over here for the two samples that we have. Okay, I have got a query to repeat whatever we did from this browsing of folders. So we are trying to see whatever output has been generated. Before visualizing it using the next tool mentioned in the tutorial, we have stopped over here after this bash k3 3k report command. I'm just browsing through, so you can just pay attention to the screen. So I'm just browsing through whatever we having. I'll just close everything and show you once again. So you were in the user folder. In the user folder, you have output generated in this folder named CQC. So I'm double clicking it. It opens another folder. So here you have out folder which has been created when we ran previous 4 commands. In the out folder, we are having multiple outputs generated for each tool. Fast QC output is in the Fast QC folder. Again, going back to out folder, Fast P output is in the Fast P folder, and centrifuge output is in the centrifuge folder. And I just showed you the multi QC report and how to open it. So you right click on this and say open in new browser tab. So here you will see the general statistics, the summary of Kraken report. So now it's using Kraken as a template to show whatever taxa were present in your metagenomic sample, the FastP output and FastQC output. So I'm closing this. So I'm just going a little bit fast so that only one part of the tutorial is left now. So coming to the last part, that is, how do we visualize the output for metagenomics? So now go to this link. Again, we are going to create a new container now. We'll be visualizing this using a different GUI container, that is graphical user interface. So click on this. Here you have to type Pavian, Pavian, and search. After that, you can launch this container. Now, when you're launching this, you have all the data being mounted in this container. So so now I'm create container. Okay. I hope everyone is following till here. We are not editing this form. It has already been filled for us. And we are just clicking create container. It's created a pod. So just refreshing it. It has started running. So we'll open it by clicking this 3 dots, connect. Everyone has followed till here, right? So now, normally this Pavian web app which is available outside does not allow you to, you cannot access data on your server. You have to browse and upload your files. But the container which we are providing has mounted its data on this web app. So here, next to upload files, there is a second tab for user data on server. So here, if you will see, you can here actually browse up. So this is in the root folder right now of your Docker image. So if you go up from this folder, you will see the drive folder which we have mounted. So double click on drive. In the drive, you have your public and user folder. In this, again, you will see all those files from the previous tutorial. So basically, in this web app, you can access whatever folders you have already created in your home drive. And then we have to go to CQC folder and the out folder and centrifuge folder since we are looking at metagenomics data. Now, if you haven't followed this in the tutorial, I have given you the path to this folder. You can just copy this path, copy, and in the Pavian tab, you can go to user data on server and paste it over here and say enter. So you will be in this same location. So now I want to see the metagenomics output. So we are selecting using mouse 1K report. So this is in the Kraken format, which is required for metagenomics. So we are selecting one file, but when I select second file, the first is unselected. So hold down your control button and select both the files and click on read selected directories. Now it has loaded both the files over here. Now to see the overview, you go to the left panel, results overview. And here you can see how many reads were mapped on the indexed file for centrifuge. And you go to the sample over here and you will actually see the interactive Sankey plot showing you the species present in your metagenomic sample. So this will, here you can see both the sample 1 and sample 2 and their visualization. So it's an interactive visualization and you can explore this particular tool in more depth in your free time. So for this tutorial, I will be stopping here. And if you have any questions, you can ask. Okay. So after selecting the files, how I came here. So I'll just close this and repeat that again. So you have created a pod. So you have this Pavian pod running. After that, you go to user data. In the user data, I'm just typing this path. Enter. So after selecting the files, what next? So after selecting the files, read selected directories, right? Now here it is selected both the files that have to be visualized. And then I'm scrolling up to the left panel over here. You will see whatever you have to explore, you can see over here. So you go to sample on the left panel. Has everyone followed? Okay. So this was about metagenomics and the use of advanced containers. So thank you everyone for your attention. Okay, so now the NGS workshop ends here and we will be moving on to the structural biology aspect of this workshop.
स्क्रीन शेयर है क्या? क्या चेंज किया? बराबर है. क्या हो गया वो? हाँ तो एक सेकंड वहां पे आया. अच्छा डिस्प्ले सेटिंग हाँ देखिए ठीक है. So good afternoon everyone. So now in the morning session and the talk by Sucheta was basically on the genomics aspect. Now we will be moving on to the molecular simulation or structure aspect. In the morning session, Tina and Neeraj showed you about the Klebsiella pneumoniae, how to identify the variants in that variant in their genomic sequence, but how to visualize that on the structure and how it affects the variant that affects the structure, that we will be looking in these tutorials. So in the brief, in the 5 slides, I will tell you why we do the molecular simulations, and then we will move on to the hands-on session where we will be using the GROMACS. So molecular simulations. So the name molecular simulations comes from molecules. So we have basic biomolecules. So it may be lipid, it may be nucleotide, it may be protein. So these are all building blocks for the different molecules and the water. So these are our molecules. And what we are doing, we are trying to mimic what is happening inside our body onto the computers. And that process we are calling as a simulation. So similar to the driving simulation, or whatever you do on the for the aero simulations. So similarly, we are trying to mimic the conditions which are present in our body onto the computers, and that's what we are calling it as a computer simulation, molecular simulations. So if you go in the literature, most of the time these two terms are being used thing. So it can be molecular simulation or molecular dynamics. So these are the two terms. So why we do the molecular simulations? So given a sequence, in order to identify the structure, if there is no structure available for your sequence, so you can get the structure for your sequence using molecular simulations. Or if you have a structure and you want to do the binding studies for your ligand. So suppose this is your ligand and you want to study where in the on the protein, where in the cavity of the protein, whether it will bind. So that thing you can study through the molecular simulations. So either this is the one example I have given for the protein folding and drug binding. There are several things where you can identify cryptic pockets in the protein, where you can understand the dynamics of the proteins, what is the effect of the mutation on the proteins. All that things you can study through a molecular simulations. So molecular simulations helps you to understand the time dependent movements of the molecular systems. And it works on the Newton's second law of motion, that is the force is equal to mass into acceleration. So it works on this principle. Now, how actually molecular dynamic simulation works. So this is in very basic nutshell. So suppose if you have two atoms, atom 1 and atom 2 at time t0, and if you apply a force on them, so they would be vibrating. So what would be their position after time t plus 1? So that's what you are trying to determine. Similarly, these are your start position. You apply initial force in the form of velocities, and once you apply the velocities, so you get the next potential. From the potential, you integrate these equations. So these are all mathematical equation, and from that you get new velocities, and from that new velocities you get a new time. So this way you are trying to understand the time dependent behavior. So whatever is happening in your body, so it occurs in the time frame of the seconds. But you are today also nobody is able to do a second long simulations. So people are generally following ensemble methods where they try to simulate multiple simultaneous simulations, and from that they do the advanced analysis where they combine the trajectory data to get some kinetics out and thermodynamics from that simulation trajectories. Now, if you look at basic simulation protocol, so this is a very basic simulation protocol. So you have initial coordinates. So from where you get the initial coordinates, so you have a PDB database. So from that you can get your initial coordinates. If you don't have a structure in the PDB database, but you have a sequence information, so now you can predict your structure. So there are various web servers available, various tools available like AlphaFold, iTicer, which can help you to predict the structures. So you can get the initial coordinates. Whatever initial coordinates you get, they may have a steric clashes. So there might be some unwanted context or something. So that's why you minimize your system. So that helps you to release the steric clashes. And whatever coordinates you have from the initial PDB, you add solvents and other ions which are present in our body, and you try to mimic the pH conditions. And because of that, the steric clashes arises, and so you try to minimize the structure and remove the initial steric clashes. And then you assign the initial velocities, that is acts in the form of temperature. So you are heating a system, so you try to bring it to the normal body temperature. So your system is initially at a random temperature, so you try to bring it to normal temperature. That's what we call as a heating or NVT simulation. And followed by you try to bring it to the constant temperature also. So where you call it as NPT or equilibrium dynamics. And then you do the final production dynamics. So once you do these things, what you get is your dynamic system. So this is one of the protein simulation what we have done for the RDRP protein, and here you can see that the ligand is binding. So these studies you can do through the molecular simulations. You can try to identify where the protein is, where the ligand small molecules are binding, and how it affects the structure. This is one of the study what we have done. One of most commonly used softwares for carrying out simulations are GROMACS and NMD. These are open source softwares. And the commercial softwares are Ember and Desmond. Now we will move on to the hands-on. So, yeah. Hmm. 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 So if you have a knowledge about the binding cavity, okay, active site or binding cavity, you go for the site-specific cavity. If you don't have any idea that where my protein or ligand is, where on the proteins my ligand is going to bind, so you take the entire protein as it and you do the blind docking for that. Now, from the morning session, you have learned how to log in on the ICE cloud. So once again, we will be doing all the hands-on session on this ICE cloud. So why ICE cloud is helpful for the molecular simulations and all that things? Because if you log in on any of the HPC cluster, you will see a black window, complete black window, and all the commands you have to type and do all the calculation. But you will not able to visualize anything. Because the visualization is generally not supported on the HPC cluster. But on the ICE, we are supporting the visualization along with the plotting software and everything. So I think everybody, all of you have already logged in. So I will do my login. So once you log in, so here you can see several tabs again, home, storage, container, HPC, and all that things. So we have made some specialized containers. So in the morning, you have looked at two types of containers. One was the command line terminals. So whatever the NGS experiments you have seen, that was a completely command line. Suchita told you about the advanced container. Now we are going to look at the GUI based containers. So that provides you because if you are a Windows lover, so whatever systems we are using, if you are working on the Windows operating system, so the GUI, whatever GUI thing, you will love it, that thing. So that's what. So here you go on the container images, and if anybody has any problem in any of the step, please put on the chat box. We will stop there and we will start over there. Okay. So once you have login, go to the container images, and here you type VMD. So you can see there are two containers for the VMD. The first one is only for the visualization. If you have a tool, if you have a small molecules or a protein, and if you want to do a visualization, so this container is for that. And this VMD GROMACS, so instead of opening multiple containers again and again for separate for the visualization, separate for running GROMACS, so we have combined everything in together and packed, and we have provided it as a VMD GROMACS container. So what you can do, you can just launch this container. So everybody from the morning session has launched so many containers. So now it would be an easy task for you to launch the container. So just click on the launch. Now, no need to change anything except the memory allocation for the image and the CPU allocation for the image. So based on your restricted usage, what we will do, we will be doing, we will be giving here 8 for the size, GB, 8 GB, and the number of CPUs, what we will be allocating 4. So for the molecular simulation, 4 are very less, but for the demo purpose, we are using the 4 CPUs, but generally we try to use more than around 40 or that many CPUs. Here we are using only 4 CPUs for this purpose. And already your path has been already added over here. So no need to do anything. And you can create the container. So your pod would have been successfully created. If you have any running container, so please delete it. Once it's showing running status, so you can do connect to your container. So you can see that every time you open the container, previously you had a black terminal window, but here you have a GUI. So it's a GUI based container. That's what. So just to repeat the steps, uh, go on the container, type vmd-gromax. So you will get a vmd gromax container. There you have to give memory allocation 8 GB and the CPU allocation 4, and keep rest of them like that only. Create on container. Your container will be created. Now you will be getting this window. I hope everybody has got this window. So we can move on to the next step. Huh? Huh. Yeah. Now, what we will be doing, so here you can see there is a one arrow sort of a button over here. So click on that. Now this is a sort of a GUI container, and what we will be doing, so we have a half screen. So instead of that, for the display setting and all that things, so it provides various options. So we will go for the full screen mode over here. And back. Now you have a full screen, so you can have, because it will be providing a better view for the visualization and everything. Everybody up to here is clear? Now, the next thing what we will be doing, again, we have to move on the black, because you are dealing with the softwares which are basically Linux based. So you have to use the terminals only. So here in the GUI, we will be opening a terminal. So here you can go on the menu and just type here terminal. So you would be having this made terminal. Just click on this and you have a one terminal open for you. So what you have to do, just I will show you the step again. Go on the menu option, type terminal. So once you type terminal, here you will be seeing a met terminal. Click on that, a terminal will open. Okay. Now, when you do the this is a Linux shell sort of thing. So there are few commands in the morning. From the morning you have learned. So ls is just I'm showing you. Ls is to list all the content what is present in this directory. So if you type ls, so you will see what are the different folders or files present in that thing. If you do pwd, it talks about the present working directory. So it's your present working directory. So you are currently in the /config. Now in the ICE or anything cloud, when you do login, you would be mostly in the folder which is specific to that container. If you close this container and if you have any files in this container, that would be lost. So always keep your files in your /drive/user directory. So even though if you close the container or pod, your files would remain as it is, they won't be lost. So just to show you that pwd talks about the present working directory. Another command is cd. Cd is to change directory. So these are the three important commands which we will be using. So cd, pwd and ls. So these are the commands. Okay. Now, in the morning, you saw that when in the ICE, you have a 3 directories. Your private one is the public, and one is the shared drive. Okay. Now, just what we can do, we can just see ls-drive public. Okay, and ICE. So just type ls-drive public and do ls. So you can find that we have the same whatever directory you were able to visualize there. The same directory are over here, and public and ICE workshop, and then molecular simulations. So this is your tutorial directory for molecular simulations. So just do the ls. We will copy in your drive. Just I am showing you the some of the files here. So I have pasted all the commands which we'll be using in this tutorial in the command.txt. These are some of the files or .mdp files, whatever .mdp files are there. They are input files for running your simulations for different steps. As I told you previously that we have to run minimization, then heating, that we call as a NVT. Then we do the equilibration, we call it as NPT. We will come again on this, but just I am telling you the why these files are required. This em.mdp has all the parameters to run the minimization. NVT file has all the parameters to run the equilibration step number 1, where we are bringing the system to our body temperature. NPT has all the parameters to bring it to the normal pressure as a temperature condition. So it's completely mimicking, bringing your system into the equilibrium condition. And finally, we will be doing the dynamics, that's we call as md.mdp. So these are the files which would be required. And this is the file called as PDB file. Okay. So this is the coordinate file which has the coordinate information, means XYZ information. We will be seeing what this file has, but it has the XYZ coordinates, which talks about the position of every atom in the 3-dimensional space. Okay. Now what we will do, we will be going to each of us, we will be going into our personal home directory. So cd drive to your user directory. So I hope now if you do the PWD, so you are into the drive user. So everybody should be in this directory currently, /drive/user. I hope online participants are also on the /drive/user directory currently now. Now what we will be doing, we will be just copying that molecular simulation folder into our home directory. cp -r. R is for the okay. So I have pasted the command to go into your personal user directory. So once you are into your user directory, what you will be doing, you will be typing this command. I will copy paste in the chat, cp-r. R is for the recursive. So in the morning, you have seen the cp command to copy the single file. If you want to copy the entire folder, so you have to give option R. So it will be doing recursively copying this thing, /drive public ICE workshop molecular simulations and dot. So this is the command to copy that directory into your home directory. Huh? This will copy all the entire folder into your home directory. Now if you do the ls, you will be seeing that folder into your home directory, molecular simulations. Now just do cd molecular simulations and enter. You should have all these files in that folder. If any of you you don't have these files in your folder, just let me know. कौन सी ये ठीक है, ये नहीं होनी चाहिए. हाँ हाँ, वो रन करने के बाद आएगी. ये फाइल होनी चाहिए एक. तो आप एक मेरे इसमें तो है. These files you should have command.txt, mdp files, this em.mdp, ions.mdp, md.mdp, nvt.mdp, npt.mdp, and this pdb file. These files you should have in your. मैं रिमूव कर देता हूँ बाकी. हाँ, सब कुछ है उसी फोल्डर में. So if you just मैं मैं तो देख रहा था ये फोल्डर दिखता कहाँ है. पब्लिक है. नहीं नहीं नहीं, पब्लिक में मैं बोल रहा हूँ. वो मेरा फोल्डर किधर है? ड्राइव वाला. मेरा फोल्डर किधर है? ड्राइव वाला. बैक है. हाँ ठीक है ठीक है ठीक है ठीक है. एक 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 ड्राइव. ओके वन सेकंड. ठीक है ठीक है. ये ओपन कर लेंगे ना अभी. इनको सबको कमांड्स रहेंगे. उनको कॉपी करने को नहीं ये पूरा उनके उसमें आ गया. वो उनको मिला, वो बोल उनको डाउनलोड ओपन करके रखो. हाँ, मैं बोल रहा हूँ वही. Now what you can do, just I will show you again. So now we will be looking at the PDB structure through VMD visualization. So before that, what we'll be doing, we will just see the files through the browser also. So just open this. There might be some folder on your this thing abd.home. So just click on this, and then over here, so you would be having slash folder. Click on this slash and then go into the drive user, and you have this molecular simulation folder over here. If you everybody has reached to this folder, or should I show again? If you click on this commands.txt, you will be having all the commands which we will be using in our this thing. So you can keep this file open. Huh? Everybody? Okay. Now what we will be doing? Just click on this VMD. Again, on the home screen, you have VMD. Just click on this VMD. So you will be having this 3 windows open. VMD main, VMD open display, and one is this terminal. Now file, new molecule, browse. Then dot, then drive user, molecular simulations. फिर से बता रहा हूं. Browse. ये मेरे इसमें यही आएगा अभी. So if there would be a drive user folder, if you click on double dot over there, click on this drive user, molecular simulations. Then you will be having this PDB file. Click on this. OK. Load. So you can see that PDB over here. This is your PDB file, which we are going to simulate it. Okay. Now here different colors over here represent different atoms. Okay. So red is for the oxygen, blue is for the nitrogen, the cyan sort of color is for the carbon. In this PDB, what we have done. So in the morning, through the variant calling, uh, all the analysis, it was reported that PMRB is the protein on which we have mutations. Okay. And क्या था drug का नाम क्या था colistin. Colistin binds to this protein, and all the secondary mechanism happens in the cell, and that leads to the death of the bacteria. But due to this mutation, mutation in this protein, resistance is conferred to this colistin, and we want to see how this mutation, single point mutation, there is only change in the single amino acid, but it affects the entire structure. So this is the dynamic change. What is going to happen in this protein? That's what we are going to see. So how a single change point mutation affects the entire structure, and that affects the binding of the colistin, and thereby leads to the resistance. Okay. So this is the PDB. So initially we did not have this PDB. So this has been predicted by AlphaFold. PMRB is a membrane protein. Here we are not simulating entire membrane protein because it would be huge protein. We are just simulating the extracellular part of the membrane protein. 1 minute. So this is the extracellular part on which we have a mutations. So we have taken only that part which has the mutations. Okay, this is just to visualize. It does not have hydrogen. Okay, but our simulation, whatever GROMAX for simulations we are going to use, they take care of adding if you have a missing atoms, not residue. If you have a missing atoms, missing hydrogen, they will add it to it. So we will see after once we prepare our system. Okay, we will again visualize. This is just to show you that this is our initial PDB. Okay, and how it looks like. So there are different visualization model. If you try graphics, representation, again I will show you on the main VMD main, you have a graphics. Just click on this, then you can create on the representation. And in the representation, you have down 2 tabs. One is the coloring method, another is the drawing method. Instead of lines, if you select instead of lines, if you select new cartoon, you can see now the structure. So this shows that it is a helical and beta combination of helical and beta sheets. Okay. Should I show again? Okay, I will tell you again how to just graphics, representation. In the VMD main, you have a graphics representation. There you have 2 tabs, coloring. If you click on this representation, you have coloring method, drawing method. In the drawing method, initially it was a line, so it looks like this. Now if you change it to new cartoon, it will look like this. It will show you what secondary structures you have in your structure, protein. Okay. Now we will just close this VMD thing and we will move on to the GROMAX thing. This was just to show how the protein looks like. So you have terminal already opened over your end. So you can again open that terminal and just do ls. Just I will show you. If you look in the text format, how this PDB look like. Don't need to you don't have to open this file. Just I will show you. So this is your PDB file, how it look like. it tells you first column is about the atom. Okay, it's a numbering, serial numbering. So you have a serial numbering. Then it talks about atom, what type of atom it is. So it's a nitrogen. It belongs to which amino acid, which chain it belongs, and what is the residue number. As I already told you that we have taken truncated protein. We have not considered entire protein. So you can see the residue numbering is starting from 1110. These three columns represent their X, Y, and Z coordinates. So that define where in the space that atom is being placed. So this talk about the X, Y, and Z coordinate, and these are the occupancy and the temperature factor. Okay. So generally we are more interested into these columns. Okay. So where we talk about the atom, amino acid, chain, residue number, and XYZ coordinates. So you can see that 1110 is entire our aspargine, then leucine, then threonine, then proline. This way you can see that this is a long PDB structure, around 135 amino acid long PDB structure. Okay. Up till now, anybody has any questions? Online or offline? Any questions? हाँ नहीं नहीं अभी तो सिमुलेशन शुरू कहाँ किया है. Okay, this is just to show you where now what are the steps to run the PDB. So now coordinate file is the prerequisite to start the molecular dynamics simulations. So either you can take from the PDB database, so that may have X-ray, NMR, or cryo-EM structures, or you can do the homology modeling, or you can use the online servers like AlphaFold, iTracer. Okay, we have already copied the data from the molecular simulation to our personal drive. So this command we have already executed. Now all the data we have in our personal drive. Now just type this command cd/drive/user molecular simulations. Already commands में है. This is a command we have to type. I hope everybody has done this thing already. Just to be sure again. Now type pwd to be sure again. So you are in /drive/user molecular simulations, because if you are in this directory, then only your simulations means, because all the file input we are going to read from this directory, and we are going to write output also in this directory only. Okay. Now the first thing what we are going to do, so we have a PDB file. Okay. Now Gromax does not understand PDB file for running the molecular simulations. So we have to convert that PDB. Yes. Huh? This is apoprotein. Docking we will be doing in the next session. Okay. Why we are doing apoprotein? Just to tell you, because whatever mutation you have, if I just replace the amino acid, because what I have done, I just replaced that amino acid, what was the originally present, I have replaced with some other amino acid. So it won't lead to any structural changes, because I have just replaced the amino acid. Only side chain and that thing would be changed. Now I want to understand what is the effect of the mutation on the structure. Okay, how that mutation is affecting overall structure, because of which the binding site or cavity which was originally present in the upper structure has been changed. Okay. Amino acid. So apoprotein acts as your control. Okay, it acts your control, and the mutant system is your protein under study. Okay, then you will do a comparative study as compared to APO. What has happened into the mutant structure? Is the dynamic has increased because of the mutant? What is the structure after 100 nanosecond for the APO structure and for this mutant? So you do the comparative study, and then you do the docking in the APO also, your normal wild type also, and in the mutant also. And then you check how the binding affinity in the APO, means your wild type, as compared to the mutant has changed. That you check. Is it clear? Yes. Now, Gromax does not understand PDB structure directly. So we need to convert it to the Gromax compatible format. Okay. So this is the command. So anything in the Gromax starts with the pre-command called as gmx_mpi. It tells that we are running the Gromax command. And this is the command to convert PDB to the Gromax format. So it's a PDB to GMX. This is the command which we are going to run. -f stands that we are giving input our PDB file, and output is our GRO file. That is the Gromax compatible file. Is it clear? Just what we will do, I will paste this command in command line also. This chat also. No. So we will be running this command in the terminal. So you will be having this terminal, made terminal, which we opened. We did the PDB. We are the molecular simulation directory, and here we will run this command. You can paste this this is your command. It should start from gmx_mpi pdb to gmx-f name of the PDB file and output file. When you press enter, it will show several force field. Okay. Now, just in a brief, I will tell you what is the force field. Now, when you are converting into the Gromax compatible format, you need to provide the connectivity information, that which atom is connected to which atom, what is the optimal bond length. Because when I am applying the force, the bond length may get disturbed to bit long. So all the parameters I need to provide. So what is the optimal bond length? What is the optimal angle between the three atoms? What is the dihedral angle between the four atoms? What are the non-bonded interaction? What is the radius of a particular atom? What is the charge? What is the mass? All these informations are stored in the force fields. Okay. And the equation that I told you, force is equal to mass into acceleration. That was a small equation, but in the background, there are big potential energy function equation. So there you compute bonded and non-bonded interactions between your different atoms or between different residues of your protein. So experimentally, so based on several proteins and carbohydrates, people have derived different force fields. So that has given what should be the optimal values for this bond length or bond angle. So they have calculated and they have put in this different force field. Okay. So these are different force field. So based on if you are whatever proteins you are studying or something, so you can do a literature survey that which force field suits best to your protein or your system under study. So for carbohydrates or for lipids, generally people prefer charm force field or amber force field. If you have to do some protein simulation, long simulation, you can go for the Gromax force field. So here you have a different options. If you go for different packages like Amber or Desmond, they provide very limited number of options. But the Gromax provides all the different force field options. So that's why Gromax has become most popular and it is an open source. So here we are going to select the force field number 6, Amber 99 SB ILND protein. This force field we are going to select. So just type 6 and then enter. The 6 is the number of that force field. So we will type just 6 and then enter. Now, when we type 6 and the enter, it asks for which type of water model you need to have. Okay. Again, similar to force field, we have, because our protein inside our body is also present in the water environment. So there are different types of water models also developed based on the charge, how the orientation should be of the two hydrogen with respect to oxygen and all that things. So there are different water models. So these different water models are compatible with different force fields. So here we will be taking tip 3 water model, which is most compatible with the amber force field. We will select one and then we will do the enter. So once you do the enter, it will complete something. It will at the last it will write some comment, and when you do the ls, what will happen? 3 new files will be created. One is called as postray.itp, another is called as topol.top, and third is called as grow. Okay, these are the 3 files which are being created. What is inside these 3 files? We will just look. Okay, just to recap what we have done. We are in the this we were in this folder. Okay, we had all these files in that folder initially. Okay, now when we run this command gmx to mpi pdb to gmx, this an output. So what it does, it takes a starting structure, it uses this command, and it creates a topology and a force field compliant structure. Force field compliant structure means your structure, now the atom types, whatever atom types are there, they are based on that particular force field. Okay, we have selected amber 99 SB ILND force field. Okay, that we have done. Here we have selected this 6 number. So we have selected type 6 to select this force field. And once you have selected this force field, next thing, you type 1, okay, to select this water model. Now, when you are working with this protein, you have several options. So if there are, now when we run the pdb to gmx command, it automatically add hydrogen also to your pdb. Okay, but if there are already hydrogen presence in your pdb structure, and if you don't want to add, so there are options you can give along with this pdb to gmx command. Ignore hydrogen. If you want to assign the n and c terminal, okay, so you can give this tar command. And if you want to choose what should be the charge, means if you are doing simulation for the protein which is come from some acidic bacteria or some, and it has a different pH condition, based on that you want to assign a different charge to this charge residues, so you can interactively assign the charge to these different residues also. These are additional options present for that. Now output would be grow file, top file, and postray.itp. Now this grow file, what is the grow file? Now the grow file, first line would be your comment. Okay, this second line in your grow file, it will be talking about how many atoms you have in your system. Okay, so these are the number of atoms in your system. This again, it has converted into the grow format. So initially it was a pdb format, you have atom type and all that things. So here you have initial residue number, followed by residue name, followed by the atom type, followed by the index, followed by the 3 coordinates, and the end you have a box. So initially this box is a means of, currently it's of no use, but we will change the box dimension because we want to put our protein into a particular box shape. And in the Gromax, all these things are in the nanometers. Okay, and in Amber, all things are in the Armstrong. This is the top file which I told you. So this is based on your whatever force field you have selected. So this top files are based on your force field. So it tells you that we have chosen this Amber 99 SB ILND force field. And these are the different parameters, means what is the charge on each of these atom, what is the mass on each of these atom, what is the atom type. So here it talks about the different atom types. So here it's the atom name, but it is the atom type. So what type? So it's based on this, it is even determined it is the sp2 hybridized or sp3 hybridized and all that thing. Okay. And this postray.itp, that column again, and this postray.itp, it will have all the numbering of your heavy atoms, and this tells you that I want to put restraint. So in the initial condition, during the nvt or npt, you keep your protein freeze and you allow only water and ions to move around it because you want to relax them first. This is the first command. Now what we will be doing, second command, So we have these three files created. Now we have created the Gromax compatible file. Now this is a second command. What we want to add, we want to make a box around your protein. So this is the command for creating the box around your protein. So again, we have to type gmx underscore mpi. Just I will paste in the chat. Paste. Okay. This is the command which we are going to run. So before running this command, what it tells that we are taking input as a grow file. We are creating a new grow file which has a box information. We are creating a centralized box with a dimension of around 1 nanometer. That means 10 Armstrong. So that much big box we are creating. It's a shape of the box is a cubic. Okay. Function. Hmm. Generally, what we try to take a box of a shape where we will try to have a minimum number of water molecules around the protein. Okay. Generally, we try to take cubic, octahedron, or dodecahedron sort of a box. So that will be having bare number of these things. The size and size of that water box depends. Means when you are applying this, because there is a condition called as periodic boundary condition. Okay, what it tries, because in your body when you have a protein, so it's not a single protein. It has a particular environment around it. Here also the same thing. Your protein, it has a particular environment around it. So in order to avoid this edge effect, so because if you are making a box of particular shape, so then it can interact only to that particular edge. It will have some edge artifacts. So you do a PBC condition, periodic boundary condition. So what it assumes that you have a similar system all around that things. Okay. So if your system moves in this direction, it will try to interact from the neighboring periodic box condition. Okay. So it won't have an edge effect. But when you apply this non-boundary interaction cutoff, so you apply 1.2 or 1.0. So your this box dimension should be such that it should not affect your this van der Waals or non-boundary interactions. So based on that, you decide this thing. So when you run this command, it will only do one modification. You don't need to open this file. Just I will show you. If you have seen initially, so initially it was 0.0, 0.0, 0.0, but now it has added the box dimension to it. Now it's a 10 by 10 by 10 cubic box. Okay. Now it has created a sort of box around your protein. Now we will be adding water into this box in the next step. So your next step is adding water around the protein box. So you are what you are doing, we will be running this next command. This is called as solvent command. Okay. So this will be adding a water around your protein. Now what will we do? We will be doing just we will be visualizing this thing. Now what you do, just on the terminal, just type VMD on the terminal, VMD followed by the name of this new box file. Sorry. This one. VMD R256G sol.grow. And if you see that, now you can see that your system has protein along with the water around it. Okay. Now just go on graphics, representation. Okay. तुमने वो कमांड रन किया, gmx solvate water wala gmx, this file will be generated after the command gmx solvate. Okay. Now just type vmd and the name of this file, and if you open, you will be having this file open. So this is to cross check that your entire protein is inside the water box. Okay. So no part of your protein should be outside the box. If you do graphics, representation, instead of lines, if you do new cartoon, okay, only protein would be visualized. Then you have to, again, we have done the similar thing. There is a one option called as create representation. Just click on this create representation. A new representation would be created, and here it would be all. Instead of all, just type water. And here, new, instead of new cartoon, just type these lines. So you have this protein. So you can have different representation. Now your protein is in the ribbon shape, and this is the water molecule. If you want to see only protein, you have to switch off this. You will be able to see only protein. If you want to see water also around it, just click this and this will be working. This is just for a visualization. Okay. Close this VMD window. We will be moving on to the next step. That is to neutralize our system. So we would be adding ions to our system. Generally, whenever you are doing simulation, your system should be in the neutral format. So overall charge on your system should be neutral. Okay. So just we are neutralizing the your system. So there are two commands for the neutralizing. First is to create a combined file. So here what we are doing, we are giving a parameter file called as MDP. We are giving a coordinated file. We are giving a topology file. We are giving a topology file. Topology file has all the information regarding connectivity and all that things. And we are creating a combined file called as TPR file. This TPR file has the information about all these three files: ions.mdp, this grow file, and topol.top. So it's a binary file which has information about all these three files. So we are making a combined file which is generally required for running the GrowMax simulations and all that things. So, but here we are creating this for adding the ions. So this, we are running this command. Okay. Once you run this command, it will, then you have to run this next command where actually you will be adding the ions to your system. So here what we are saying, that if there is a positive charge on my system, you add Na ions. If it had a if it has a negative charge on my system, add Na ions. If it has positive charge on my system, add Cl ions to make overall system neutral. Okay, this is my this command tells me this thing. When I paste this and run, it will ask me that it has to add ions, but there is a limited scope. So I need to replace where it has to add ions. So what I will say, that remove 3 or 2, whatever number of ions you are going to add. Here it is telling that it will add 4 sodium ions. So there was plus minus 4 5 charge on my system. So it will be adding 5 sodium ions. So I need to replace some 5 atoms, different atoms. where it can. So I will select water sol 13. Okay. So it will replace 5 this water molecules with the 5 solvent atom. It says that it has replaced this solvent molecule number this with this, so on. Okay. Now, if you look at your topol, you don't need to run. Just I will show you. So if you check your topol.top system at the end, what you have one protein molecule, you have 35,400 and water molecules, and you have 5 sodium atoms. Okay, this is overall your system now. Now your system is ready for simulations. Okay. So all whatever steps we have done till now were for the system preparation only to start the simulations. Okay. Now what we are doing, we will be actually running energy minimization. Okay. Now what we are doing, we are again creating a combined file called as em.tpr. Here we are using a parameter file called em.mdp, then this sol ion.grow, topol.file, and we are creating as a combination of these three em.tpr. Just copy this command. Now just type ls. This file would be created em.tpr. Has everybody got this file em.tpr? क्या एरर क्या है? तो जी निकाल दो थोड़ा सा. वो पिछली फाइल में हमने थोड़ी गड़बड़ हो गई. What has happened? Because in the previous command, we have created the file. There we have wrongly written, instead of R256G, we have written R256 only. And what command I have given? I have given here G. So just remove this G and your work will be done. I hope everybody has got that file em.tpr. Online people also, I hope everybody has got this. If anybody has any issues, please tell me, because the next step we are going to run simulations, energy minimization simulations. Okay, now here what we are doing, here we are doing, we are running now parallel job. Okay, so that's why whenever when we have created the container, we have asked for force. अभी पूछो मैडम, दिख रही है क्या स्क्रीन? उसको बोल दो कि जस्ट रिफ्रेश करेगा वो. Now we are going to run a parallel job. So here this command, this says that we are going to use MPI run. So it's telling that we are using -n4. So we are telling that we want 4 processors to run this job. MD run is the engine to run molecular simulations for the GROMAX. And here, instead of big command where we are talking about what input and output, we are telling default only that by name of my input file em.tpr, it is em.tpr. So I'm just telling that my name of my input file is EM. So it will create all other files, means output files with the name EM dot something extension. Okay. So when you run this command, just copy and paste. It's doing minimization. So it will calculate force on each and every atom. Okay. So you can see that energy minimization step is going on. It is calculating force on each of an atom, every atom. Wherever the force is maximum, it tries to apply some. नहीं, हम लोग क्या कर, दो चीज करते हैं उसमें. हर एक मतलब ग्रोमैक्स में जनरली हम लोग स्टीपेस्ट डिसेंट, मतलब एक एल्गोरिथम का नाम है स्टीपेस्ट डिसेंट. ओके. उसमें हम लोग क्या करते हैं, नंबर ऑफ स्टेप्स स्पेसिफाई कर देते हैं कि वी हैव टू रन फॉर दिस नंबर ऑफ स्टेप्स. ओके, दैट इज वन क्राइटेरिया. एंड सेकंड क्राइटेरिया इज दैट if the force on any atom is below this 1000 kcal something, then you have to stop. कोई इशू नहीं है, ये अच्छा एक ये एक अच्छी चीज है. ये जो हाँ, ये जो हमने एडवांस कंटेनर यूज कर रहे हैं, इसका भी हमारा टेस्टिंग हो गया है कि जिसने जॉब रन किया था उसका जॉब किल नहीं होगा. बाकी जो आप कंटेनर यूज कर रहे हो, जैसे ही अगर इशू होता है तो वो कंटेनर में जो जॉब चल रहा है दैट गेट्स किल्ड. ओके, बट हियर द जॉब यू आर कनेक्टेड ना.
Yeah. Enter the refresh. You just refresh it.
जस्ट रिफ्रेश, आपका जॉब चल रहा होगा. So this is the beauty of the ice. So this was for the testing purpose that your job is running or not. So here actually this is the advanced container. Here we have a given provision just for the online users. So here the network got lost and we were not able to get connected to the ice thing, but our job was running in the background. So here you have these two windows, ice cloud and this thing. If you don't kill this terminal, okay, if you don't close this terminal and if you close only this, okay, if I close this, again what will happen, if when I connect, I will go to the same window. Okay, so my job is, if it is running, it will be in the running state only, so it won't get killed. This is for the beauty of this advanced container. I hope everybody's minimization job has been completed. Online users also. Till now, till this step, if anybody is facing any issues, online or offline, anybody? Okay. Now we will be doing our next step just to check whether the convergence has or not for our simulations. So this step in this, whatever potential energy calculation has been done. So here, where if you do the ls, so ls-lrt, so this different files have been created: em.tpr, em.grow, em.edr, em.trr, and em.log. Okay, this different files have been this was the input, this is the output, final output of this energy minimization. This is the energy file which has the log for all the, this is complete log file, and this is the trajectory file. It has a time series data sort of thing. We will look into it also. Just now, what we will be checking, we will be checking the energy, whether it has converged or not. So there is a command called gmx. Sorry, gmx-_mpi energy. This command, if you run, it will ask for which parameter you want to check. Okay, for which parameter you want to check whether the convergence has happened or not. Here we will select 10 for the potential energy. वो just एक. Now when you do ls hyphen LRT. So list. So this potential.xvg file would have been created for you, everybody. Potential.xvg. What we have done, we have run this command gmx underscore MPI. It would have given you several options. Just press 10, then enter, then enter. Here you will get several options. 10, then enter, then enter. Okay. Now we will see, we will do the plotting also. So now in this advanced container, we have visualization also, plotting software also, and simulation software also. Just type XM Grace. XM Grace is the plotting software. Potential XVG. You can see that after this number of steps, it has got saturated. Okay, this shows that your system has converged. After around 1700 steps, it has got converged. So this shows that it is reaching saturation after this many number of steps. So your system has energy minimization has been completed for your system. So you can now move on to the next step, where what you will doing, step equilibration. Okay, so it is called as NVT. Here what you are trying to bring your system to the body temperature. So you are having constant volume, constant temperature simulations for this. Prior to that, we will just we have to do one more step called as creating an index file. Why this index file is created? So suppose now we have only protein, but if you have protein and ligand systems, okay, in that case you have to club that both this group, that protein and ligand together will act as a single system. So here just we will be creating this index file. You don't have to do any, just type this command and then Q, press Q. Type this command and press Q. So it would have created an index file for you. But if you have ligand also in your system, then you have to type here, suppose protein and your ligand system was 13. So you have to type 1 pipe 13. So this will make one common group for that thing. So here we have only protein. So I have not done anything. So it has created for me an index file. Now we can go for the next command. That is called as creating an NVT TPR. Means again we are creating a combined file called TPR, where we are combining NVT.mdp, em.grow. Whatever output we have got from the last step, we are taking that and topol file for creating this thing. And so now it has created for us TPR file. Again, we will run the simulations. So generally for NVT, we give more number of CPU cores, but here we have a limited choice. So we will be giving 4 CPU cores only, and we will run this NVT simulations. Okay. Now it will take time for everybody of you. So in the meantime, what we can do, just here file, open terminal. Okay. So it will open a new terminal for you, and we will just look at what has happened to EM file. So you type vmd em.grow. So whenever you have done the minimization, so this thing, and then here right click on this, load data into molecules. Here we have to, because this trajectory file does not have the connectivity information, that information it will take from the grow file, and you will load the TRR. So it was a minimization. It has 2 frames only, so you won't see much dynamics. But once we complete this NVT thing, we can see some dynamics here. Just the basic small dynamics we might see, not much. But once we complete the NVT, we can see these things. So NVT will take time. In the meantime, what we can do, we can go to You can close this here from the top, so we can have some docking or some Jupyter Notebook tutorial. So once this is completed, we will do the visualization for this NVT run. Just close from the top. Don't close the terminal. Huh? Okay. If anybody has any doubts online or offline, you can put on the chat. For the neutralization, what we are doing, so you can see that what we are doing, we are creating one common file by combining ions.mdp, this grow file and topol file. So we are creating a combined file called as ion.tpr. Once this tpr is created, what we are doing, if you don't know what is the charge on your system, you are telling that if it is a negatively charged, add sodium ions. If it is positively charged, add Cl ions and make it as a neutral system. Make it as a neutral system. That's what you are telling. So it will create a file which would be having ions added according to your system. Okay. Till the time this NVT simulation runs, what we will having some docking tutorials by Shruti. Let it run. Don't close this thing. Shruti?
Okay, am I audible to every? Okay. So now I'll come to the home page again. Now we'll be having a brief like an exercise on the molecular docking. So mainly molecular docking is nothing but you have a receptor molecule or the protein or the biomolecule under consideration and a small molecule which you want to check the binding with this particular biomolecule, that is a protein. So currently, as since morning, we have been talking about AMR and the initial analysis which was done before lunch, we came to the conclusion that TMRB, one of the proteins, might be the one that binds to the antibiotic colistin. So, and a mutation in this protein only leads to the antibiotic resistance. So in MD simulations, we looked at the effect of this mutation, like what conformational changes it might occur. So now what I'll be going through an exercise where we dock this to the wild type PMRB and maybe the entire set is given to you. The same you can replicate for the mutant later on. So for that, the docking software that we are going to use is Doc6. And again, here it's an advanced container. Why? Because we all know in structural biology, as you saw in MD simulations also, many a times we had to interface with GUI, where we had to come and check in VMD whether things are happening right or not. So same holds true for the molecular docking as well. So for viewing, in case of molecular docking, we'll be using the Chimera, UCSF Chimera tool. And for docking, we will be using the Doc6 tool. So for that, we will be loading a new container. Now you do not delete this container because your Romax is running already. So don't touch the this particular container which is already running. The one that is running over here, kindly do not touch this. We'll create a new container. So just go to container, images, or I would suggest go to the storage drive first. And in the public drive, hope everyone is with me. In the public drive, in the same folder that is ICE Workshop, there is a folder called as molecular docking. So here in hands-on, you have this PDF. Kindly download this PDF. So the initial few instructions are the ones which you have been doing since morning. So you might be acclimatized with this, and I know that you are already logged in into. So you can directly start from the page 3, where I request you all to create a new folder. So I'll just tell you how to do it. In the storage drive, go to the file drive. Now this is your own drive. Now just right click over here. Sorry, create a new folder. There's an icon over here, new folder. Just click on that. And I request you to name it as for, so that we are on the same lines. You name this output for this folder as moldoc_tutorial. These instructions are also there in the PDF that we just downloaded. Here I have mentioned. And just say create. And you get this folder created. So now in your home, you have this particular folder that is moldoc tutorial. So now what we'll be doing is we'll be performing all our operations on /drive/user/moldoc tutorial. Okay. So hopefully this is done. This is what you can refer, the PDF that has been attached. I'll wait for a few seconds so that everyone has created. Okay. So now we'll load the instance for the molecular docking. So go again to container tab. In that, images. Just in this search bar, type doc and press on the search icon. You get these 3 containers over here. We will be using the third one, that is Chimera-doc6, the GUI container. As I said, for molecular docking also, in between steps would require visualization. So this is the container that we are going to load, and just say launch. Again, being an advanced container because it has multiple things. Here, whatever your private drive is, that is what we were calling as /drive/your username. Now that will appear as /drive/user. So if any user is there, it will appear as /drive/user only for all of them. So there won't be any confusion based on username. So again, here it's same. We are kindly do not change this job name and image name. Do not touch them. And again, here for memory allocation for this image and CPU allocation for this image, I request you to do 4 for the memory allocation and 4 for the CPU allocation of the image. Rest all, you need not change anything. And say create container. It is saying starting the container instance. Okay. So successfully the pod has been created. And here I can see it's in the waiting status right now. And if I refresh it. Has it loaded for the ones who are here? Yeah, like now we are want to check the mutational effect. So only a single amino acid change in the question asked here is whether we should go for simulations first or molecular docking by the audience here. So in case of mutational studies, it's always better that you do the simulations first, because what conformational changes might occur because of the introduction of that mutation needs to be captured. If you have an experimental structure of the mutate, then you can directly start docking first and then perform the simulation with the docked molecule. But in case you do not have a mutant structure, it's better that you mutate and then you perform the MD simulations. And after a considerable time, you start the equilibration of the MD simulations. Later on, a production run for some time that it shows some conformational changes with respect to wild type. Then you select a ensemble representative from your mutant, and that you can dock and again perform simulations. That should be the ideal strategy for studying a mutant. Okay, it's provisioning now. We are getting closer. It seems we all are tired after the whole day, so the ice cloud is giving us some rest. So mainly here, as we are talking about this PMRB, so what I'll be doing here is for the wild type structure, I'll show you the docking. Maybe you all can try for the mutant structure also. So we have kept the mutant PDB for you. It is not, but it's not a simulated PDB. It's just a replaced PDB, but just to have an exercise and to see whether you get any changes. Yeah, we can. There are Discovery Studio tools. Toolkit has it. Chimera has it. And if you're using Amber tools, there is this Leap programming, T-Leap, X-Leap. They all, if you just keep the backbone atoms, you just replace the name in the PDB and load it. It redoes it with the new residue name and adds the sidechain according to the residue name. You should remove all the sidechain atoms. Just keep the backbone atoms and the 3-letter amino acid code in the PDB. Okay, so hope everybody has got running state. I have got it for me. So now here in actions, again, we say connect. Okay, so completely new tab again opens up. The tab name is moldoc-doc6. So I request everybody to just stay on this tab henceforth and not go to another tab except for the PDF if you want to have a look. Okay. So if you look at the PDF, we have finished these steps. We are currently on page number. Yeah, we are on page number 7. This is how it looks. Okay. Here you might get this message. Kindly say allow if it asks you. So our first step here is mainly in molecular docking. What happens is we do our docking exercise in parts. First is the receptor is a protein molecule. So you might download it from PDB. So the PDB structures doesn't have hydrogens and doesn't have partial charges assigned to it. But all this is required because finally docking is nothing but an energetically favorable binding of the small molecule and the protein. So the charges and the hydrogen atoms all need to be in place. So the first thing what we are going to do is for this is whatever files for the tutorial we have made, it has been placed in okay, for that first go to menu and say it says ask some message, say allow. Type terminal here. Again, here we'll require a terminal similar to the previous here again. So here you are on your terminal, I think so. And if you want to go to the folder that you just made, then say cd/drive/user moldoc tutorial. This is your folder where you will be performing all the exercises. This has been written over here. You can just copy this and say cd and copy this path. Okay, so now here you are in your folder. If you type ls, which lists the files, currently you don't have anything because this is the new folder that you created. So we are going to get all our tutorial files into this, the inputs. So for that, again, if you go to the PDF, you can see in blue color, we have highlighted the link, the path for all the files that are present. So including the excluding the inverted commas, copy this particular line and say CP, right click, paste, space, dot, star, that is all the files, space and dot. And then again, if you do ls, all the files are here. These are the input files that we'll be needing. This doesn't have any of the output files. So here is a small description about what the inputs we are going to use for this particular tutorial. So this PMRB wild type is nothing but the wild type PMR protein. And this particular mutant, which was also taken for studying in the MD simulations, this is the same, but with the mutation R256G. For small molecules, there are various formats like PDB, SDF, MOL2. So what we have done here is this file, you can directly get it now also from the PubChem itself, because for small molecules, we refer to the PubChem database itself, and we can save that over here. So currently we have provided it, but if you go and search through PubChem, that also, you can keep that one also. INSPH is one of the inputs that is required for the future steps. I'll talk about each of this whenever they are coming in the due course of the tutorial. So currently you are in this molecular mol doc tutorial. Okay. You have copied this, all the files. So we have completed till here. Hope everybody is with me. Okay. So now we have the files in our drive in our own location. So if you want to just go on the other tab and see the storage file drive and look at the mol doc tutorial, you have all these files now in your own drive, in the user drive. Okay. So come back to this mol doc6 tab. Okay. And the first step for molecular docking is the preparation of the ligand and the receptor, that is adding of hydrogens and the charges. Is there any doubt? Excuse me, sorry. So now is where our GUI part plays a role. So first we will, as I said, our receptor currently will be this PMRB underscore wild type dot PDB, and our ligand will be colistin underscore mol underscore 2D dot SDF. Why 2D? Because many of the molecules are not present in the 3D format in PubChem. And for colistin, when we downloaded it from PubChem, it was in the 2D format. So we have also kept the features in this particular container itself, wherein these ligands can also be prepared. So accordingly, we'll go through the step. So now I request you to minimize this terminal. Whatever you have minimized lies in this lower bar over here, what you can see. So just double click on this chimera. This window will open. Okay. Now we go to file. We want to open the wild type PDB. Open. Now here you can see this is the base folder of our container, and we know we have to go and drive user. This will be same for everyone. There will be no different name here. It would be the word user. And inside this small doc tutorial. Once you have selected this, or keep this camera open, it will always be in this particular. Otherwise, if you accidentally close it, see to it that you come to this path. And here we have this PMRB underscore wild type dot PDB. And you just say open. With the left click of the mouse, you can rotate this. So you can just visualize this. You can increase this window. The middle scrolling this in the mouse can be used for zooming in and zooming out. So this is how our wild type is, what we have selected currently. Then we go to tools. If you look at, yeah. Okay. I'll start again for loading of the PDB. I'll close this. Double click on Chimera. Okay. Go to file. Open. Our base folder is drive. In that we have user. And in that we have mol doc tutorial, which was created by us, the folder. And in this, we have the PMRB wildtype.pdb. And say open. And maximize this window for better viewing. Okay. So now when we look at the steps we have performed, we are now This is the path. If on page number 12 of the PDF, you can see where we have actually placed now copied our PDB. So we can load it from here. So this is what we see. Now the first step is adding of hydrogens to the receptor molecule. So it says click on tools, then structure editing, and the first option itself, that is add H. Just click on this. So here currently only a single PDB is loaded. If there are multiple PDBs loaded, they all would have been listed here. So this is a single PDB. Checkbox this only, and here you say unspecified so that all the residues are considered, and just say okay. So here you can see on the lower left bottom, hydrogens added had appeared. So now hydrogens have been added to this. Again, now the next step is, so you see the same, it's been given here in the PDF. You can have a look if you have missed it. These are the selections that were made. They have been highlighted, whatever selections have been made. Hydrogens have been added. And the next is assigning partial charges to the receptor molecule. So again here, click on tools, structure editing, and just the second option, that is add charge. So here again our PDB is selected. For the standard residues, there are different force fields that are used for adding the charges. We will use the latest one, that is the Amber FF14SB force field. And for any non-standard residues, currently in our PDB, there are no non-standard residues. But sometimes there are PDBs which carry these magnesium zinc ions based on if there are DNA binding proteins also. So there are certain ions that are present along with the PDB. So at that time, this particular charges may be used, methods may be used to add the charges. Just say apply and okay. Here again, it says total charge added. So now the partial charges have been added on our PDB. But what the PDB we have doesn't have all these added. So now we need to save this so that henceforth we'll be using this. But we won't be saving it is in the PDB format. If you look here, after assigning this, now we save it, we will be saving in the mol2 format. So just go again, file, and you have this option of save mol2. Click on this. You are, because it's the same window opened, you are already in the same folder. That is your current directory, that is moldoc tutorial. And here you just type PMR capital B underscore wild type capital underscore charged dot mol2. This name has been written in this file. You may copy it from here itself. This is the name. You can just copy this and say save. So here it says wrote this particular file in this path. So this is only our path, drive user, moldoc tutorial, and this. Hope everybody is with me. Any questions till now? Has everybody been able to save this mol2? Okay. So now our receptor has been prepared for further exercises of docking. Now what we are going to do is we are going to prepare our ligand. For that, currently just minimize, do not close it, just minimize your camera and again come back to your terminal. Keep it maximized. And now if you do ls minus lrt, it will give this new file that you have created, that is pmrb underscore wild type underscore charge dot mol2. So you have this now. Just I'll clear the screen by writing the command clear. So now my next step is preparing the ligand. So my ligand is, if you just type CO and press tab, the entire file will come because this is just a single file with this name. So this particular file SDF is with me. So now for this, we have one more command line tool called as Open Babel, which performs these operations on small molecules. As this is a 2D molecule, we require a 3D molecule for our docking studies, and also we need the hydrogens and the charges added to this. For this, if you come here you see this command O Babel. O Babel is nothing but Open Babel. Just copy this command. Control C over there and right click and paste over here. So this is my command to prepare. So here what is it? That file type, the input file type, that is SDF, the name of the input file, hyphen O, the output file type, that is mol2, hyphen capital O, name of your output file. So I have just renamed it from 2D to 3D and the extension has been changed to mol2 as this is a mol2 format. And this hyphen gen 3D is the flag that will convert it into 3D. And hyphen partial charges is assigning of the charges. We use this Gastiger method for assigning the charges to this particular compound. Just press enter. This might take few minutes. Till then, we can go on the further steps. So if everybody is completed till here, you can just minimize this window and come back to our Chimera window and just open it. So we can go back to the Chimera window. So now here is, we need to now prepare the receptor molecule. Now, based on the surface of how this particular molecule is, we need to define what might be the probable binding site for the small molecule that we want to see. Because currently for PMRB, there is no structure available. So this is a predicted structure that we got. And that's why the binding site is not defined. So in such cases, what we do is, in case of rigid docking, what we do is, we generate a surface around the protein and then we pass on a spear to fill the gaps which come anywhere across on the surface. And the one with the maximum, and this is filled with small balls, you say, or spears. So more number of spears, larger is that particular cavity. So what you do is you select the one with more number of spears. So when you do not know the binding site, the largest cavity that is in the protein that is obtained by surface calculations is the one that is selected for binding, in case you don't know it. Yeah. Yeah. Yeah, if you get those, then you can provide there is a provision in this to provide the residue names. Around that, it will create the cavity, because this is a place where we don't know about the cavity, so we are doing this approach. There are two more approaches. One is site specific, wherein you exactly know, means there is already a structure with a bound small molecule. You give that also as input. So wherever it has bound, it will just take that cavity to dock your new molecule. And the third one is this residue specific, where you provide the list of residues. If you give that, then it will create a cavity around those residues to do the docking. So currently this exercise, we are doing the blind. This is called as a blind docking, because we don't know where. We'll just select the largest cavity. So for generating the surface, this is the command. As you can say, go to this, you open this Chimera, go to actions, surface, and say show. Wait for a while. This is the surface on the protein created. So this is the surface information. If you scroll through, you can see now there are certain dips, again, I'll hide this. So this was the state you were in, everybody. So you go to actions, surface, show. This gets created. Here again, I have marked here also, the highlights have slightly got misplaced, but it is, I've written here action, surface, and show. Okay. So now here you can see there are certain valley kinds of things. If you look here, there is something like a valley. So this might be a cavity. So like this, now this surface information helps to identify the cavities. So how do we capture this surface information is we write it into a .dms file. So here again, if you go in this tutorial, go to tools, structure editing, write DMS. Tools, structure editing, write DMS. Here again, we in our folder. Now here again, we have given the name. So we'll name it as PMRB underscore wild type. I've just kept the name consistent so that we know that, sorry, it is the same file. Sorry, DMS is molecular surface. It is a file format for molecular surface as we just showed the surface on Chimera. So to save that surface information, DMS is the type of file format. So maybe it is not getting copied. So just see to it that you write this only. It is nothing but PMR B underscore wild type underscore surf dot DMS. And make sure you're always in this file, this folder only what you had created, and say save. So here it says wrote DMS surface. All right. So just minimize it and we'll come back to our terminal. Okay. So here again, now if you look at the terminal, you see here the command that we had run for the ligand preparation. It says one molecule converted. So your file now, this cholesterol underscore mol underscore 3d.mol2 is your prepared ligand, which you will be further using for docking onto this PMRB wild type. So now again over here, what you can do is our next step is to now get these, make this surface into that spheres. Like we have to now calculate which is the largest cavity. So for that, you just do more INSPH. There is a file with this name. It will get displayed. So as you see, this is the file that we just created. This is an input file. This we have provided in the tutorial. This was the surface information file that goes as an input. And this will be the sphere file that will be generated. Okay. So the command, you can look at the PDF also I've written here, more INSPH, what is get displayed. And then to run this, to make this sphere, simply type SPH gen. It's written over here. You can copy also SPH gen. You can see here. And just press enter. It's complete. All right. So now, as I said, this would be our output file of this SPH gen. Just double click on this, copy, more, paste. Just check what is into this. So here you can see it is a huge file. And here you can see this cluster 1, number of spheres in cluster 94. So these, what you have selected right now, like the largest cavity, they could spot 94 spheres. We'll be visualizing this also. I'm just showing you what is this .sph. So if you just press space, you can walk through the file and you can see cluster 4 has come. So there is cluster 3, cluster 2. So at different locations, it might get cavities. So these clusters are nothing but the different locations all over the protein. So just press Q to come out of this more command. Okay. So now here, we can select this just single cluster and make a PDB of it. The first cluster, which in this case had 94 spheres. For that, the command has been like, if you just do this more, this is nothing but the input to select that first cluster. So here PMRB wild type.sph is our spheres, what we just saw, this file, the more what we had done. One is the first one. N is yes or no for generating writing the surface again. So we are saying we don't want to write the surface again in the PDB. So we say no. And this is the PDB which will have those 94 spheres only which are there in cluster 1. So for this, the command to make this PDB is show sphere and the input file sign. You can see here and the SPHgen cluster.in. You just need to place this and you say enter. So here see the first cluster which had 94 spheres has been saved over here. Okay. So if you just want to have a look at this, is everybody with me till here? So just now click on this chimera. We can now see where these spheres actually are. Okay. Now in order to see the spheres, we need to get rid of this surface. So just go to actions again, surface and say hide where you had said show first. Just say hide. So you'll get this representation. Here now we have to just open this PDB along with this. So file, open. We are in R folder only, and here we had created this PMRB underscore wild type underscore spheres.pdb. Right? You say open. You see here a few spheres are generated here. Dots, you can say, they appear as dots. So this is one of the cavities that has been selected for docking. This is the one which was the largest cavity that has been, so we'll be, okay, this file name is, I'll show again. I won't open it again. This is PMRB underscore wild WT underscore spheres.pdb. If you just check the PDF, this has been shown here also. We have given here, it's the same name. Okay, so this was just to show you that where this cavity is. Now minimize this window. You're back on your terminal. So here, after this, what you would need to do is, because we want just the first cluster, our input file our file that was generated, this PMRB wildtype.sph, just copy it to one more file named PMRB underscore wildtype selected.sph. So just take this command, right click, paste it. Okay. So now, because what we are going to do is, in this new file, we'll be keeping only that cluster 1 information and remove everything else. So wherever your line, this cluster 2, this line starts, which you can, how you can see it is, do VI and select this, copy, paste, open. Now this is like an editor, it has opened. Okay. I'll go back again. So you have copied your PMRB underscore wildtype.sph to the selected. It's the same file, but this new created file we will be editing. That's why I made a copy. So you just open in your VI editor. Now just type slash. You can see in this portion over here and write cluster. So this cluster word gets selected. And if you keep pressing press enter. Okay, so you press and then press N, you come to this line, or you just scroll through the arrow marks and come to this line, cluster. What I want you to do is, including this line, the rest of the lines, I want you to delete it. And just till this line, this should be, this 4003, this should be your last line. Okay. So how you can do that in basic Linux operations is, just press escape now and see that your cursor is on cluster 2. Press D, Shift G. नहीं हो रहा. Okay. It's done. Or what you can do is just, okay, D Shift G, it kills all the line, and then you say again escape, colon WQ, exclamation, this will just save your file. So basically you have to save this file is what I say, that after from the line cluster 2, nothing should be there in your this file, because this will tell the further steps that please use the largest cavity, that is the cluster 1. Okay. So that's why I mentioned here the things might change. So I've just given you this cluster 2 wherever it is. Okay. Okay. So now what we'll be doing is the next step is called as box generation. So we need to enclose, need to provide an enclosure inside this cavity around which the new protein, a new ligand will be docked. So for that, I will just do clear so that the screen is clear. And the next file is box.in. So again, if you see here, yeah. Here 5 mentions the dimensions of 5 Armstrong for the box. And this is the selected SPH that will go as an input. And this would be our box, that is output. Okay. So here also if you go, more box.in, we did that. And the next command, okay. I'll repeat the last 2 steps. So what we did was this PMRB underscore wild type.sph, which was generated through SPH gen, we copied into a new file, PMRB underscore wild type underscore selected.sph. Okay. And then we opened this in VI editor. Those who are familiar with Linux, this would be easy for them. And here I am on this first line. I come to the line where either by page down also you can come. You can come to the line which reads cluster 2. You can do page down and come to this line. Once your cursor is on this line, just do D shift G, D shift G. So you have to do it fast. So it deletes all the lines. Or if you're not able to do that, just do it by pressing D, it will finish. Just it will take some time. The file is not generated then. Okay, the SPH gen command you haven't run then. Whoever doesn't have this file, just run the command SPH gen. That's it. You had generated the .dms file, right? You know, see. Whoever is getting stuck, kindly follow the instructions in this PDF, because this is how we are sequentially running the steps. Okay. So now we had done this more box.in. So now our next step is creation of box. So our input would be the selected sphere and our output would be this box PDB. So for that, the command is given here, show box, Control C, right click, paste. Okay. Press enter. So it has been run now and finished making the PDB format box file. So now again, if you want to view this, I hope everybody has got this. This is run successfully. 94. I'll just minimize this and open the camera to visualize this box. Again, go to File, Open, and here this PMRB wild type box.pdb. Those who have, they can have a look here also. We have given the snapshot over here. User, Moldoc tutorial, and we are in PMR B wild type box.pdb. Just select this and say open. Now you see it has enclosed these peers, this box, what we can visualize. Okay. All right. So this is how we have created the box. Now minimize this again and come back to your terminal. So the box has been generated now. So after this box generation, it is time to make a grid around this box for more specific docking of the small molecule. For that, our input file is this grid.in. So here you say, just copy this more, paste grid.in. So this is the input file. So here you can see, you can check these files by doing ls. Just double click this, say copy, ls, paste, enter. So yes, this is present. Kindly check each of these files by copying, paste the file and do ls. Here again you can see these both were the files that we created, if you remember. This is after the addition of hydrogens and partial charges to the protein, and this is the last step output, that is the box. But you can see here one more file over here. If you do ls and try to view this file, do you see this? Yes, you see this. Now this is something what was pre-installed in this advanced container. These are the files which are provided by DocSys. These are the parameter files for performing the energy calculations. Because as I said, energetically favorable binding has to happen. So for that we have van der Waals parameters, extra electrostatic interactions. So whatever are cutoffs and the parameters for the what atom types they are there, all that has been provided by DocSys. So this is like the path of where the DocSys has been installed in this particular container, and it will always remain the same. So we have this, you need not change at all, because this is the DocSys. If you're using it in this container, this is the DocSys that you're using. So even if you haven't made it, you haven't copied from anywhere, this file exists. That is because this had come with this container. Okay. So I'll just clear the screen, and the next step. So I have written here, it is the system file which has come with the container. So you have to type this command now, grid, copy this, paste this. -I grid.in, -O grid.out. Enter. So this might take a few seconds. Does anyone have any doubt till this particular step? Because this is the penultimate step before we do the actual docking. Because till now you saw we are treating only the receptor molecule. Up till now, after the ligand preparation, we haven't. Yes. Charged. So after you, in the first two steps, when we added the hydrogen, we added charges to the protein. Then we had to save as mol2. Just check that. You can, if somebody doesn't have the .mol2 file, I'll just tell them what they can do once again is they can again open a fresh chimera. Okay, so that we don't disturb this one for the future steps. Open a separate chimera, file, open. Again, you have to go to our user directory, the Moldoc tutorial. In this, kindly load our first file, open, go to tools. Sucheta, you may follow this now if it is not there. It's there? Okay. Follow this tools again, and you can keep this and save it again. Like you can do structure editing, add hydrogen if required. If any of the file is missing, if somebody has forgotten to save the mol2 file initially, they can do this thing. Addition of hydrogens, addition of charge. Now just file, save mol2. And this is the file that we had saved. You can replace it the same name. I won't say do this saving, but you do this and save this file if at all it has not been saved. Okay. So now here the grid.in is running. It will finish in a few more seconds, I guess. So what you can do is after this exercise is complete, you may you may use directory. There is this particular file, pmrb underscore r256g.pdb. You can repeat the same steps that has been given in this PDF from the start, and you can do this exercise and check whether there are changes in the results. It is running. Till then, we can just, till this is happening, come to the main ice cloud page. Yeah, where is, close that, yeah, you know, go to the instances and we can go and have a look at the GrowMax, the VMD GrowMax pod which was there. You may connect it and check whether the NPT is completed. Here, if you're getting this screen, the password for this is ABC for everyone. Just write ABC and unlock. This is nothing but a screensaver. It is still running this particular so okay, we'll come back to this molecular. This is also still running. Okay. So maybe I'll have a sip of water till this is done.
अरे उसके नीचे परसेंटेज भी नहीं आया इसके लिए मैं देख रहा हूँ इधर 10 20 की
नहीं है।
इसमें नहीं आ भाई
हो जाएगा और okay, till that time we'll just have a look at the next step. For that, what you can do is here in file, you can open a tab in this terminal itself and you are in the same folder. We'll look at the next step, that is the actual doc6 command where we'll be giving. And if you look at the tutorial also, you have to do this more rigid.in. So this is for rigid docking. As I mentioned earlier, we do not know the binding site. So we are performing a blind docking and rigid docking exercise. Just say more rigid.in and press enter. So here is where our molecule will come into play. The small molecule, we'll give the name of the molecule. This is the selected sphere. That is the cluster where in the cavity in which we need to do the docking. And here, as I mentioned, these are the different parameter files that are required for calculation of the energy. And here you can see something called as ranked ligands. And we have said yes. Why is this used is in doc6, we can not only give a single molecule to doc, but we can also give a database of molecules to doc. Like we have a single mol2 file, which is a concatenated file of multiple ligands. If we give that as an input, what it does is it docks each one of it and ranks them according to the grid score. So if you want that ranking, you have to say yes. If you say no, then alphabetical order of the name of the ligands that you have given, it just gives you the grid scores. You just look at the other tab. Why is this step so lengthy? Is the box that we have generated over here, in order to fit in the molecule, it is forming a grid across this box where the actual ligand docking is happening, and this grid will happen such that there are no steric clashes between these protein residues as well as the new small molecule that will be loaded. So I can just show you, there is a, just type this command, I'll paste it in the chat box also later. Like what is the output like? So I'll just show you. Drive public. I'll paste this link, don't worry. 1 second, I'll just reach there. So this is the command. I'll paste it in the chat. I've pasted it in the chat. So if you have a look at this ls, so here there are few file names with the starting with grid. So these are the different out files, except for grid.in. These are the out files that will be generated after this successful execution, and it's done. So this grid command is completed. So here only, if you just press ls minus lrt, if you type, you have got these grid commands. Okay. And we already had a look at the rigid.in. So again, you can have a look and just check whether these files are present or not. I'll just open this file completely. You can scroll above. So the first file required is this cholesterol. Hope you have saved this. This has been saved through that obabel command that we did. So ls paste. Yes, it's present. Next input file is this, copy. Yes, paste. This is present too. And these are the doc6 parameter files, which are by default present in this doc6 container. So they should be present. It is always better that whatever inputs, if input links you have provided here, you cross check, because sometimes we don't realize and we are not in the same path where these actually lie. So yeah, all these files are present. Okay. So I'll just clear the screen and we'll fire the final command which will perform the docking exercise. That is doc6, which is again given over here. You can directly copy, paste. This command might be time consuming. Right now it won't be, because it is just a single molecule that we are dealing with. But when we have seen there is this high throughput molecular docking, something what we have heard a lot. So there what happens is there are databases of these molecules that are docked at a time for two multiple proteins. So at that time, what you require is parallelization. So here is where parallelization plays a role. That's why this doc6 is a serial version. There is a doc6.mpi2 command which is present that can be used for parallel execution of this, for faster execution also. So this will give us a rigid ranked.mol2. Currently that will be having only one molecule, because our mol2 file had just one molecule. This will finish now. So on the other tab, the same link what I had pasted, you can again copy and okay here. So here you see this rigid out and rigid rank dot mol 2. So I'll just show you how it looks. If I do more rigid rank dot mol 2, I'll place, sorry, this is not here. So I'll do this ls command, I'll play again, and in front of this command only I'll paste this. I'll paste this command again. This is the pre-generated output that I'm showing you. Anyways, once our output is generated, we'll have a look at that too. I'll just paste this command on the chat. So here you can see there is this grid score, grid van der Waals energy, grid ES energy. So this name is nothing but this is the CID or the PubChem ID of this particular molecule. Number of rotatable bonds in this molecule, heavy atoms, the molecular weight of cholesterol, and here the formal charge on this ligand, number of hydrogen bond acceptors, donors, heavy atoms, and this is actually the grid score or the dog score, what we say. So this is what we refer to when we want to see how strongly it has dogged or not. So the more negative, the more better this value, the more negative this value is, the more better binder that particular molecule is. Just have a look. Okay, so it is done over here now. Come back to your the old terminal where this doc6 command had run. Just do ls minus lrt. So you have this rigid dot out and rigid rank dot mol 2. So in order to visualize this, where is that ligand dog? Go to this chimera here. Go to tools, surface binding analysis, and view doc. Here you can see this has been given the entire tools, surface binding analysis, view doc. Okay. So here what you have to upload is this rigid underscore rank dot mol 2 is what you have to select and say open. So now here it asks that this output has been generated from which docking software. So we have used doc6. So we'll select this particular option and say OK. So the same information is now displayed over here. So how much? Minus 44 is the grid energy that has been seen here. The same information has been, of course, now you would say that pre-run, what happens is the number of clusters that are generated at that time, based on that, this grid sort depends. So currently, this is what we have got. So if you want to see the location, just say hide to this. This tells you the grid score and just say hide. And now you see the one in ball and stick. This is where your ligand has been docked. For more clarity, what you can do is go to tools, general controls, model panel. This is not a part of the tutorial. This is just few features of Chimera that we can use. So just waiting here. Tools, general controls, model panel. Here you can off the other things. Just keep the PMRB wild type. You can just off these views. So you can now clearly see where it has docked. So this is the location where it has been docked. And if you wish to save this structure into a single, now you had to do the view doc and everything to save this, to see this. But if you want to save this particular structure, just go to File, Save PDB. And here it says multiple files. You say it as single file. And here you type as PMRB underscore wild type dogged dot PDB. Okay. So this will be a single file and say save. So now if you open this, now if I go and off the visualizing of this for some time. Now this is empty. File, open. This PMRB wild type underscore doc.pdb, open. Now it shows you this, right? So now as a PDB, this has been saved. Okay. So this was the molecular docking exercise. So what you can do is, instead of the PMRB underscore wild type, you can use the PMRB underscore R256G.pdb and perform these steps. However, whatever naming you do in the files, accordingly you change in the .in files and the INSPH file. And you may just compare, like here for wild type, you got the grid score of -44, what you get for the mutant. And that way the difference, if it is more negative, which shouldn't be the case ideally, because the mutant is not letting the colistin bind. So ideally it should be greater than -44 is what is the ideal scenario we should get. So this is about the docking essays. So this PDF is at your expense. You can use this for okay. If we check this, VMD it's still running. So here, what I request everyone is, hope everyone has understood there are no more queries here, but I understand as this was the first time, you might have some queries. So we are open to it, and we have this on the home page. If you click on home on your ICE login, ICE Cloud login, here in the feedback, if you just click on feedback, here you can fill this feedback form and submit so that we can know what all is more required from your side, as well as how did you totally feel, because you didn't have to do any prior installations for running any of your tools. And give us a review about this particular workshop. How did you feel about?
करो करो Yes, sir. Yes, sir. Yes, sir. Can you ask?
What I feel is, you may type. Please fill the feedback form. That's the one thing. But currently, we have some time to take some questions. So you might just type it in the chat. We'll address them. It can be what are your requirements also, and what, and it, as we had mentioned earlier, that this logins will be available, like you can use this for 1 week, like this data, the logins will be available only, but this data, whatever you have done, will be available for a week from now. So you can use, there are certain tutorials. If you have a look at the help page over here, there are certain tutorials given for variant calling. We'll be updating this also. So you can have a look at this. And anyways, right now you have the PDFs with the instructions which are at your expense. You can use them. And you can give us a feedback also on what are, because as in when you use, you might come to know what are things that might be more can be included into ICE Cloud. And kindly share your feedback after performing few more activities on ICE Cloud for this 1 week. That
And this ice cloud is available from anywhere, accessible. It's not that you have to use it in your lab only. You can access it from anywhere, from your home, from your mobile. You can submit your job. You can check the status of the job, visualize any molecule, or we upload data. So that is what is a big advantage. You can share your results with your guide or any other scientist. So that is also big plus with this. See, you have done something, you have saved it, and then it is now given to the, just you have to share with your guide. Can see it and he can give feedback, comments, all those things.
Audience present here, you have
And
some.
there is a email ID also given for queries.
It is also there on the ICE website. You can see here. You can use these email IDs for your queries.
Their audience में अगर है कुछ issues, then we can address. Otherwise, anyway, it's you can try on your own now. Workshop is over as such, but you have to try on your own. Okay, and next 7 days, and even after that, if you really come up with a big scientific problems, then we are there to help you through ICE. Okay. So it's not that 7 days is just to learn, but
Okay, how
later
can I
on,
use?
if you really want to run very long simulations or analyze particular data sets, then we are there to help you.
For Python code development, we have this VS code also. If you see over here, there's one query. I'll just read it out. How can I use for development code on Python? How can I import libraries? So here, yeah. So here in instances, sorry, in images, if you see, we have this Jupyter Data Science, Jupyter TensorFlow, Jupyter R. So these are actually Jupyter notebooks that have certain pre-built libraries. But here, once you launch this, I'll just give a small demo. You need not change anything of this. If it is a heavy memory job, you can accordingly assign memory over here and create container. Visioning. Okay. So here on the right side, we have these. Just copy this password, actions, connect, paste. Paste this password, say login. So here you are in your Jupyter notebook. So here is the Python kernel you can open. And here you can import the libraries you want. Like in data science, we import this pandas, numpy is used at a large scale. So these are already existing. And if you want to install some, here itself you can give pip install and the name of your library and run it. And you can use this for your Python coding. And while saving this, here you can see, like this is Vinod's logging. So this is my /drive/user name Vinod. This is the public drive which is read only. So you can read input files from here. You can't write anything into this drive. So what you can do is, while saving this, you can save it inside this also over here. So this is where your local storage drive is loaded. We can directly see it from here itself. Means you can have your input files here and you can load it here and do your analysis using Jupyter Notebook.
Acha, input level. In future, we can use services for free.
In future, we can use the services for free or it's going to be paid up.
For academia, anyway.
For academia, okay. The next question is, in future, can we use this service for free or it is going to be paid afterwards? We are yet on deciding that, but currently the academia will be provided it. We'll be providing it for free.
See, academia and R&D will be, government labs will be free, research labs. Only for private people, I think it will be charged.
And currently, there will be a basic quota that will be decided for each of these, whoever want to use the users, and that quota would be assigned, and that much you can use then.
And it won't be in dedicated mode because there will be multiple users. But if somebody has any project requirement and if they can pay some compression charges, then definitely in dedicated mode also we can provide this facility.
Does that answer your question, ma'am?
Okay, now you are aware of Ice Cloud and it's available to you now. We want you to try out. You tell your colleagues. You can they can also try not just MD and docking and maybe genomics, but Python is another. I mean, not just Python, but R. Even you can do programming as such means. There are Docker images available. You can write here code, compile it, and run. Not just Python, any other software development also you can do. So that kind of facility is available. So we want you to try out. So this is the first one. So it will be definitely given for access. Okay. Any other query or any other inputs, any other feedback? If otherwise, we can close this. Surita, do you want to say anything? Yeah, you can directly load your Python script.
PS code.
नहीं, वो पूछ
For coding purposes, we have this VS Code environment. Over here, you can see. So here, most of the coding tools, Jupyter also we can use, and this also we can use. If you have your PY file, as I mentioned here, you can place it in your drive, and if you open it from here,
private drive.
in your private drive, that is /drive/username, inside that, if you place your .py file, you can open it here and you can do the coding. For example, I'll just
One question more. मेरे इसमें मोड़ो था.
so okay. So here I just made this notebook here, XYZ. So even if this is your outside notebook, if I close this now, so here in this my folder, I have this xyz.ipynb, and in my instances, I have already run this Jupyter notebook. So I can just open this again. So here, this xyz.ipny is appearing here because it is a part of my home storage. I can double click here, and here I can have this particular file, whatever I had created.
So any file you can import on
Yeah.
this platform in your private drive.
What is the maximum resource that can be requested by a user?
think it should depend upon the problems.
Actually, it would actually depend upon the problem statement, like what project requirement you have. You need to justify the requirement that you're giving. So based on the authorities here, then it would be decided whether it to be provided or not.
For academic,
Yes.
it's a free.
Yeah, Ice Cloud is free for academic users. Okay. Okay, so we'll close for the workshop. Thank you very much for your patience. Okay, suppose we have 2-3 people, how can we work jointly? So I'll just show it here. Like this is one of the logins, Sucheta, you can see storage file drive. I just this XY, this particular file, maybe go to private. My drive. In my drive, I'm going into a folder. Maybe say I want to share this doc6 folder or the MD Crow, this folder I want to share with a user. So right click, there is share. So whomever you want to share with must be registered on Ice Cloud. So based on that particular person's username. So like currently here, I have. Sorry. एक minute. Okay. ठीक है. So I'll just share it with Shruti Kaulgi. Here is whether you want it to be read in only or read and write. So if you want to do a code development, you need to give read write permissions. Currently, I'll just keep read. And here you can specify till what date also you can do. So this feature is specially for researchers wherein there are collaborations which are limited to a particular project. So this feature enables that we share the data amongst each other till the project completion timeline. And later on, the data stays, but the sharing is removed. So the one who was the owner of the data is having the data then, not the ones with whom it was shared. So I can just select here. So currently till tomorrow, if I say and say share. So the file is shared. So you see a new icon appears over here which says it's shared, which is not in case of others. So I'll just now open one more tab of Ice Cloud and I'll log in into my account. So here again in my, if I go to file drive, I see shared, here say one user shared file with you. I click on this. It is the same folder. You can see the MD Crow. So I can now access this particular folder. It was this folder, right? So this is how sharing is enabled over here for fruitful collaborations. Okay, so we'll end now.
Any other
Thank
feedback
you
from
very much. Hope to
physically
see you
present
using eyes in future. Thank you so much.
Yeah, you want to still explore it and understand the potential of Ice Cloud.